What is this? What is this?
A DNA strand like me.
Ladies and gentlemen, welcome back. We're discussing here today a special topic, DNA sequencing. What is really meant by DNA sequencing? DNA sequencing is a process that enables us to determine the accurate and exact order of DNA sequences. It is simply done through specified methods that help in the determination of the 4 bases of DNA. DNA sequencing techniques are of great importance in the fields of medicine and biology. Identifying DNA sequences aid in the process of research, especially in biological fields. Further, specific fields where DNA sequencing is of extreme value. Firstly is the medical diagnosis. Secondly is the virology. Thirdly is the forensic medicine. And fourthly, the biotechnology. The process of DNA sequencing is divided into 3 major steps. The sequencing reaction can be repeated for about 30 or 40 cycles. The first step is the denaturation, which is done around temperatures of 94 to 98 degrees Celsius, in which the double strand melts to obtain single-stranded DNA. The second step, annealing, is done around temperatures of 50 to 65 degrees Celsius. The single-stranded DNA or RNA pair by hydrogen bonds to form a double-stranded polynucleotide. The ionic bonds are constantly formed and broken between the single-stranded primer and the single-stranded template. The term is also often called renaturation. The third and final step is the extension, which is done at 72 degrees Celsius. In this step, the DNA polymerase synthesizes a new DNA strand that is complementary to the DNA template strand through addition of dNTPs. After the sequencing reactions, the mixture of strands has to be separated. This is done in an acrylamide gel. This is achieved through gel electrophoresis. The DNA that has a negative charge migrates to the positive pole and vice versa. The smaller fragments will migrate faster than the larger ones. The fluorescently labeled fragments that migrate through the gel are being passed through a laser beam at the bottom of the gel. When the laser exits the fluorescent molecule, it sends out a light of unique color. That light is collected and focused by lenses into a spectrograph. Based on the wavelengths, the spectrograph separates the light across a CCD camera, otherwise labeled as charge-coupled device. Each base has its own color, so the sequencer can detect the order of the bases in the sequenced gene. Each of the bases give out a specific color of its own. Thus, the sequencer detects the order of the bases in the sequenced gene. For publicity purposes, each sequence of a gene has to be confirmed in both directions. To accomplish this gene has to be sequenced with forward and reverse primers. Since it is only possible to sequence a part of a 750 up to 800 bases in one run, a gene of, for instance, 1800 bases has to be sequenced with internal primers. When all of these fragments are sequenced, a PC program tries to fit the different parts together and then assembles the total gene sequence. In 1975, Sanger method was developed as the plus and minus method. However, the breakthrough occurred 2 years later through enzymatic polymerization in 1977. The method revolutionized genomics. It is often called the dideoxynucleotide or chain termination method. It involves the use of 4 separate reaction tubes that are set up, each tube containing an identical DNA of interest, a radioactively labeled primer to get DNA synthesis started. The deoxyribonucleotide phosphate to be used in DNA synthesis as the dNTP, and a small amount of the dideoxyribonucleotide phosphate, the ddNTP, and the DNA polymerase. The Maxim-Gilbert technique, unlike Sanger, it involves the chemical degradation of purine bases. The purine bases, such as adenine and guanine, are damaged by dimethyl sulfate. Through the methylation of bases, the heat releases the bases, and the alkali cleaves the guanine. This method is based upon the detection of the produced PPIs during polymerization of DNA. The emitted light helps us determine the sequence. There are two approaches to this method, the solid phase and liquid phase. This method increases the enzyme's efficiency and yield. This is a laser-based method that allows rapid DNA sequencing of fragments at a rate of 100 to 1000 bases per second. It is based upon the detection of individual fluorescent nucleotides. This method is based upon the sequencing through synthesis chemistry. Each DNA base is labeled with a different color of fluorescent dye. The DNA polymerase produces fragments that are ligated to adapters at both ends. Those adapters are considered as primers. This method is based upon the ligation and subsequent steps. The fragments of DNA are ligated to adapters and bound to beads. The amplification of DNA fragments is achieved through emulsion PCR. The DNA denaturation occurs. We add a ligation mixture after we use a primer. I'd like to thank you, ladies and gentlemen, for your time and attention. Thank you all.